In this section, we will talk about the reabsorption of bicarbonate ions and secretion of hydrogen ions. Moving towards reabsorption of bicarbonate ions. About 4,320 milliequivalent of bicarbonate is filtered by the glomeruli every day. It is called filtered load of bicarbonate. Excretion of this much bicarbonate in urine will affect the acid-base balance of body fluids. So bicarbonate must be taken back from the renal tubule by reabsorption. Now, moving towards secretion of hydrogen ions. Reabsorption of filtered bicarbonate occurs by the secretion of hydrogen ion in the renal tubules. About 4,380 milliequivalent of hydrogen ion appear every day in the renal tubule by means of filtration and secretion. Not all the hydrogen ion are excreted in urine. Out of 4,380 milliequivalent, about 4,280 to 4,330 milliequivalent of hydrogen ion is utilized for the reabsorption of filtered bicarbonate. Only the remaining 50 to 100 milliequivalent is excreted. It results in the acidification of urine. Secretion of hydrogen ion into the renal tubules occurs by the formation of carbonic acid. Moving forward, carbon dioxide formed in the tubular cells or derived from tubular fluid combines with water to form carbonic acid in the presence of carbonic anhydrase. This enzyme is available in large quantities in the epithelial cells of the renal tubules. The carbonic acid immediately dissociates into hydrogen ion and bicarbonate. Hydrogen ion is secreted into the lumen of proximal convoluted tubule, distal convoluted tubule, and collecting duct. Distal convoluted tubule and collecting duct have a special type of cells called intercalated cells that are involved in handling hydrogen and bicarbonate ions. Secretion of hydrogen ions occurs by two pumps. One is sodium-hydrogen antiport pump, and second is ATP-driven proton pump. Moving towards sodium-hydrogen antiport pump. Well, sodium-hydrogen pump occurs in the distal convoluted tubule. When sodium ion is reabsorbed from the tubular fluid into the tubular cell, hydrogen ion is secreted from the cell into the tubular fluid in exchange for sodium ion. The sodium-hydrogen antiport pump present in the tubulars responsible for the exchange of sodium ion and hydrogen ion. This type of sodium-hydrogen countertransport occurs predominantly in distal convoluted tubule. Second is the ATP-driven proton pump. The ATP-driven proton pump occurs in the distal convoluted tubule and collecting duct. This is an additional pump for hydrogen ion secretion in distal convoluted tubule and collecting duct. This pump operates by energy from ATP. This was all about this section. In our next section, we will talk about the mechanisms involved in preventing metabolic acidosis.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.